Good afternoon, everybody. I'm Matt Parker, and me and my colleague, Surisha Hesketh, are going to show you some of our end-to-end solutions for the analysis of both TB, human, and mRNA vaccines. So both Surisha and I are registered clinical scientists. We previously worked in the NHS, delivering diagnostic results to patients, and we both know the importance of applying regulation to these bioinformatics workflows and the sequencing devices. And so we're building all of those processes at Nanopore so we can deliver end-to-end solutions in the clinical and biopharma areas. So we're going to talk about three things today. We're going to talk about AMPOR-TB, our test for the resistance of tuberculosis. We're going to talk about human variation, and Surisha's going to do that one. And then I'm going to come back on and talk about mRNA vaccine quality control. So the first one is TB. So we all know it's a massive global health problem. There were 10 million active cases of TB in 2022 and 1.3 million deaths. And only COVID-19 is a bigger infectious disease killer. There's a problem with drug resistance, right? We can't have 10 million people walking around with unknown resistance profiles, having the chance to acquire more mutations and cause more drug resistance. So we desperately need to find out what these patients are resistant to so we can stop antimicrobial resistance spreading and also tailor their treatments. Traditional methods of culture can take weeks, and whole genome sequencing isn't rapid or flexible enough. Also, other sequencing technologies cannot deliver rapid results like Oxford Nanopore can. So the WHO recently supported the use of targeted next-generation sequencing for antimicrobial resistance profiling in TB. And we participated in the FIND trial, so we did 600 samples across 3 sites in South Africa, in Georgia, and in India, and we performed really well, and the WHO endorsed our test for these applications. So we can go from sample to report in 6 hours, so it's a really limited hands-on of a preparation and extraction from sputum, PCR, library preparation, then 2 hours of sequencing, and automated analysis. We screen for 16 antimicrobial agents, and I'll show you those in the next slide, on our GridION device, which we're looking to move to an IVD. We do 22 samples plus 2 controls per run, and you can run this up to 4 times per flow cell. So that brings costs down. So we target regions of the TB genome that are responsible for antimicrobial resistance to common first-line therapies, newer second-line therapies, including the BPAL regime and bedaquiline. We also have additional targets like HSP65, which allows us to tell if you have any other non-tuberculosis mycobacterium in your pot. We also look at the CRISPR locus to tell us the Spolliger type of TB, that's like the lineage, and we also have internal controls. So my colleague, Surisha, is going to demo how this works on our GridION device. So it's really, really easy to select an assay on these devices because we've now delivered an end-to-end solution. So here, Surisha is selecting TB, and all of the sequencing settings that you need are pre-configured so that you don't have to select them and no mistakes can be made, which is really important in a clinical lab where perhaps you're all really busy trying to get those answers out for patients. So you do your flow cell check, you would load a sample sheet, and you would click go. And then eventually you get to the point where after 2 hours of sequencing and around 30 minutes of analysis, we're able to see the report. So if Surisha clicks on the report, it's right here in the software, so no additional analysis is required. It's all done for you. At the top, we have information about the sample, and then we have important quality control information. So if we click View Details in the Run Status area, we can see the coverage of our no template control. So you'll see here this includes a QC amplicon that amplified well in this case, and no other target amplified. So that's a pass for the no template control. And then we have a positive control, and that amplified well, which means we now can progress to the samples. So the assay was successful. So for each sample, we give a really clear antimicrobial resistance profile. You can see here this first sample, we were able to detect TB, and the red box there, if you scroll down, Surisha, it's the 4th one down, yeah, this one. The red box here indicates that this sample is resistant to rifampicin. So if you click View Details, yeah, that's one. We do more quality control checks on sample coverage. So the sample's got to be really well covered so that you know you've got TB in there, and we've been able to detect all of the mutations. Also, we look at the HSP65 coverage and the internal control. If we scroll down a little bit further, we can see the variants that we've detected. So in our highly accurate nanopore sequencing reads, you can see that at this position in the TB reference genome, there was supposed to be a C, but in fact, we detected a T in the RPOB gene. And this means, because we use the WHO catalog of mutations, that this causes resistance to rifampicin. It also gives the allele frequency and other information that you might need to make a decision on what you want to report to your patient. So we can scroll down further, and we get things like the coverage information, taxonomic classification. So here, if we had an NTM, we'd be able to see that. And if you scroll down further, we have spoliger typing. So that's our test for tuberculosis drug resistance, AMPOR-TB. It will be available in the latter part of the year as an RUO. And we're really excited to bring this to you guys to use to better tailor treatments to patients who have tuberculosis. So next, I want to welcome Surisha to the stage, who's going to tell you about human variation. Thank you.
Thank you. Hi, everyone. It's great to be here to talk to you about our human variation workflow. So I'm going to talk, the main part of this talk really is to tell you about a really exciting upcoming integration that we have with Minnow. But for those of you who may not be familiar with the EPI2ME human variation workflow, I'm going to start with an overview of what the workflow does, the types of outputs it produces. I'm going to highlight just one particular sub-workflow and an interesting variant category that we can detect called short tandem repeats, or STRs. And then I'll finish with an overview of what the Minnow integration is setting out to achieve, and Matt is going to show you what that actually looks like on the device. So yeah, just as an introduction to the human variation workflow, this is one of 17 workflows that are maintained by the EPI2ME team, and it's a really comprehensive, thorough analysis of human genomes. As you can see from this slide, we can do small variant calling, structural variants, we can look for copy number changes, we can do short tandem repeat analysis, and we can also analyze modified bases. And the beauty of this workflow is all of these sub-workflows are independent, so depending on the type of analysis that you want to carry out, you can choose one or all of these, and it's all part of the same automated process. So what do you get when you run the workflow? Well, they are a complete set of comprehensive outputs, all of the standard sequencing outputs that you'd expect, haplotagged BAM file, variant calls, really thorough coverage summaries, and all of the sub-workflows each independently produce an HTML report. And the standard file formats that are produced from this workflow are suitable for downstream analysis. So as I said, I'm going to talk a little bit about one specific sub-workflow. So I'm sure most of this audience is very familiar with what a short tandem repeat is, but just as a reminder, there are certain, it's a class of variant where you have short, between 2 to 6 base pair units of DNA, which are, as the name suggests, repeated in tandem. And these happen all over the genome, and in lots of cases are perfectly benign, but in certain genes and in certain regions of certain genes, too many of these repeats can give rise to a disease phenotype. So the example here is a plot from the STR report that's generated when you select the short tandem repeat sub-workflow, and in this particular example, it's a Coriell sample where this person is affected with Fragile X syndrome. So you can see that in the two alleles, the left-hand blue section there is for a length of the region which is in the normal range, and then in the affected allele, you can see the triangle over to the right-hand side is representing where the number of repeats has fallen into the pathogenic range. So this is a plot that we have in the report. We also have a second plot. This was a really fantastic collaboration with an NHS trainee clinical scientist who came to us last year and spent 6 weeks in the EPI2ME team. Her name is Sophia Johnson, and as Matt mentioned at the start, he and I are both registered clinical scientists with the HCPC, and Sophia is actually enrolled on the program to achieve the same registration. So in that 6 weeks, she did an incredible amount of work. She got to grips with the code base of human variation, and she looked at the existing plot, and we tasked her with adding a repeat content plot to the STR output report. And this was a really great addition because because the long reads that we can get with Nanopore are really great for this particular class of variant, which is why I've chosen to highlight it today. And the repeat content, as you can see, it's the same individual as the previous plot. And we have the two different haplotypes, and you can see that in one, the reads are within the kind of normal, healthy range, and in the other, they are very much expanded. And the main CGG trinucleotide repeat is the kind of blue bars, but you can see that where the interruptions occur, it's really clear to see that there are some reads where we have these so-called interrupted regions. And the reason this is really kind of clinically relevant is that there are some conditions where the presence of these interrupted regions can affect the age of onset, it can affect the severity. So this was a really nice kind of clinical collaboration that we had with this NHS trainee. So we're really grateful to Sophia for coming and spending that time with us, and we got a really nice plot out of it as well as a result. So I just wanted to say a little bit about annotation, because if you're doing human genome analysis, annotation is really key. It's not just enough to generate a raw list of variants. You have to do some filtering and have a look at the kind of consequence of these variants. So we do some basic annotation in the human variation pipeline, which is using SNPF, a tool that some of you may be familiar with, and then we also overlay ClinVar annotation. So this is the view of the report from the SNV sub-workflow, but this is also available in the VCF output as well. So again, suitable for downstream analysis. Sticking with the theme of annotation in human variation, we're really excited to announce an upcoming integration with some tertiary analysis partners. So this kind of gives you even more annotation oomph for your money. So you're going to be able to link your outputs directly with a trusted third party for onward analysis to get you even more biological context of your results. So that's kind of a whistle-stop of human variation with a sort of small spotlight on one of the sub-workflows. The main reason for doing this demo is to talk to you about the integration that we have with Minnow. We're really excited about this. This is going to allow users to queue the analysis to run immediately after the sequencing is finished. So this is coming in the next release of Minnow, and the idea is that at the same time that you set up your sequencing run, you're also going to be able to set up your data analysis. So we are really kind of taking away any of the complexities that are associated with analysis of this kind of data, which is great if you're a newcomer to doing data analysis. If you're comfortable with Minnow, it's just a small step further to set up your analysis. And this will all happen without any further interaction from you. You just set it up and it's ready to go. And it's really bringing us ever closer to the vision of true end-to-end workflows. Matt's going to now show us how this actually looks on the device. So he's just going to go and set up the run and click the box for setting off the analysis, which you can see there. At the moment, this is just for human variation. So when you select to do the analysis this way, you'll get the option of selecting the different sub-workflows that you might want to look at. And depending on what you select, you can alter the properties of the parameters that you supply. And as I said, all of this will be queued to run as soon as the sequencing is finished. So I think that's everything I will be saying about human variation. So Matt, back to you for vaccine QC.
Thanks, Tarisha. Thanks, Tarisha, for that great demonstration. This is just the start of integrations, right? We want to bring more and more end-to-end solutions to our devices because we know it's easier to go from sample to answer without having to have complex workflows after the sequencing's finished, transferring files around, selecting the right analysis. So this is just where we want to start with. And another application where we're working on end-to-end is in our biopharma area. So we have existing workflows for biopharma. We have two that are freely available now. You can download these analysis from GitHub and use them in EPI2ME. These are our workflows for the QC of adenoviral vector production systems. That's workflow AAV QC, and also our workflow for basic DNA constructs, which we call workflow clone validation. They're both available right now. They're really easy to run in EPI2ME, and you can get really nice HTML reports that show you QC metrics for both of those applications. Integrations with these on our devices will be coming later in the year. But really what I wanted to talk to you about today is a really new, exciting application of nanopore sequencing technology with mRNA vaccine identity QC. I think we're all familiar with mRNA vaccines. They changed our lives during the COVID pandemic. immunizing us against COVID-19, and more applications for mRNA technology are coming to the forefront. And really, these new amazing technologies for vaccine production need new innovative technologies for QC. So right now, mRNA vaccine QC uses a whole host of traditional methods. So to get all of this information, things like the identity of the mRNA molecule, the integrity and the purity, you need several different instruments. These are complex and time-consuming to run. You need several members of staff in the lab who have to have the knowledge on how to use these machines correctly. What we're offering is a GridION device with an integrated analysis to replace all of this testing. So right now, we're able to do sequence confirmation. We're able to analyze modified RNA transcripts. We're able to do transcript integrity to see if they're intact. And we can do poly-A tail length estimation. 5' cap detection and double-stranded RNA is coming very, very soon. So why now? Why can we do vaccine QC now? Really, there's been a culmination of several really exciting developments within Oxford Nanopore. We have really innovative sequencing chemistry with our RNA-004 kit, new flow cells, new chemistry, and also updates to our base caller. So the new version of Dorado, version 0.7, has new models that enable really highly accurate base calling of our direct RNA sequencing data, as well as poly-A tail length estimation. And also, we're making innovations in our software analysis, so we can bring all of these updates onto one device to make it easy to run from end to end. So it's really easy to go from mRNA sample to a report. You can take unmodified mRNA or modified, process them with RNA-4. You can set up some configuration and acceptance criteria for the workflow, because we understand that different mRNA vaccines will require different configuration and acceptance criteria. You would load them on a Q-line GridION and click Go, and then you would get a report, which is in HTML or PDF format. And we're working to ensure these are all CFTR Part 11 compliant for GMP. So like TB, you can just click Run Setup, and you can select the mRNA vaccine QC identity test. You would load your sample and a sample sheet and click Go. And then around 2 to 3 hours later, you'll be presented with a report. So here, Suresh showing you the mRNA identity report. At the top, again, we have quality control information. So those acceptance criteria that you've defined will be shown here if they fail. Also, all of the information you need to meet CFTR Part 11, all of the metadata required for those regulations. If we scroll down, you can see more information about basic run performance, things like the number of reads, the length of the reads, the accuracy. So actually, this is new RNA-004 data on an internal quality control mRNA molecule with a poly-A tail of 125. And you can see the median accuracy there of 96, which is really incredible. RNA-004 data is absolutely amazing. It's really impressive. And if you scroll down further, we can see more information about coverage. So one advantage of nanopore sequencing is that we get the full-length molecule in a single read, so the whole of the mRNA. And what that tells us, if we get the whole mRNA, is that that mRNA molecule is intact. If we get shorter reads, that means there's some degradation in your preparation, and that's important for vaccine producers to know. So this plot here should be shifted all the way to the right for a really intact mRNA molecule. If we scroll down further, we then provide information on the poly-A tail length. So the poly-A tail is really important for mRNA vaccine efficacy, and making sure that this has survived your production process is really important. And so here we provide, for each read, a histogram of the poly-A tail length, and this so happens to be a poly-A tail of 125, and the median prediction here is 125, so this is a spot-on prediction of poly-A tail length. So if you scroll down further, we provide information on the sequence identity. So we make a consensus sequence and compare to the reference you provided. And here, this one's perfect, apart from a couple of bases in the poly-A tail where consensus building is quite difficult. We also can predict and base call the linker sequence if there is any present. So thank you all for listening to the demo today. I hope you'll agree that there are a couple of game-changing applications in both diagnostics for tuberculosis and in mRNA vaccine quality control. So Sarisha and I are happy to take any questions. Thank you for attending London Calling, and we'll see you all soon. Thank you.